Today's lecture is going to be on the nephron loop system or the production of urine. When we're looking at the nephron loop system, there are two major nephron loop systems that occur in the kidneys. We see the cortical nephron and the juxtamedullary nephron. Each one of these nephron loop systems are all going to have the same working parts. The first one is going to be the structure called the renal corpuscle, which houses the glomerulus capillary system. It will have the proximal convoluted tubule, the nephron loop system, the distal convoluted tubule, a collecting tubule, and a collecting duct. The only difference between the two is going to be their location. So when we look at the cortical nephron, which composes about 85% of our total nephrons in our kidneys, the primary location is going to be within the cortex, even though we do see a little bit of the ascending and descending limb of the nephron located inside of the medulla. The juxtamedullary nephron, again, it has all the same working parts. This constitutes about 15% of our total nephrons in our kidneys. But the main difference is, as we can see here, is the nephron loop system is going to go deeper inside of the medulla. So just to give you a general overview of what we're going to be looking at, we're going to be looking at blood. 20 to 25% of your total volume of blood is going to filter in through the glomerulus. And this glomerulus is a capillary system. In this, we're going to be pulling out filtrate. So solutes, water, and ions are going to be pulled out. The remaining oxygenated blood is then going to leave this glomerulus capillary system into the efferent arteriole. So once the filtrate is going to be entered into the proximal convoluted tubule, it will then pass through the nephron loop system, so it will travel down through the nephron loop system, and then eventually it comes into the distal convoluted tubule. It travels through the collecting duct, or collecting tubule, into the collecting duct. It'll travel deep inside, so we're about at this level here. It'll go through the minor calyx to the major calyx, to the renal pelvis, and then eventually into the ureter. So once it's going through the ureter, then we consider the concentration no longer as filtrate, but we call it urine. So the first and most important area that we're going to be looking at is going to be the blood flow and the filtrate. And so we'll first look at the blood flow coming in. It's going to be coming in through the afferent arteriole, and this is going to enter into the glomerulus, which is a capillary system, through this region called the vascular pole. So we're going to have blood entering in through this capillary system, and we're going to see filtrates, solutes, water exiting out into the renal corpuscle or the glomerular capsule. And then once it enters into this region, then we consider this to be filtrate. The filtrate will travel to the tubular pole, and then eventually it'll go through the proximal convoluted tubule, through the nephron loop, and to the distal convoluted tubule, and then out for excretion. The blood that's traveling through is not losing oxygen, so therefore what we see here is it's going to exit through an efferent arteriole. So we have oxygenated blood coming in, solutes, ions, and water are being pulled out, and then the remaining blood is going to pass out of the capillary system into the efferent arteriole. Remembering that we have 20 to 25% of your total blood will be going through this filtration process. So we have a couple of important areas that we should also mention around the vascular pole is going to be the juxtalar glomerular apparatus. And the first one we're going to look at is the macula densa cells. These macula densa cells, which are located closer to the vascular pole, these are important because it's going to be monitoring all the ions in the tubular fluid. And what that means is that these cells are going to be connected with the tubular section of the nephron loop, and it's going to be monitoring the different types of ions. This is important because while it's detecting the tubular fluid ion concentration, if the blood volume or solute concentration is reduced, what we see is that these macula densa cells are going to stimulate the juxtaglomerular cells to release renin. And when renin is released, it activates the renin-angiotensin pathway, which then causes aldosterone, which is a hormone, to be produced, which increases your blood ion concentration and increases your blood volume. So this cell is really important with its link to aldosterone and the ability for your kidney to change blood ion concentration and to increase blood volume. Another cell that we should mention is going to be the granular cell, and these granular cells, which are located along of the afferent arteriole, these are smooth muscle cells of the afferent arteriole, and this allows for the lumen to widen, so the ability to vasoconstrict or vasodilate the amount of blood that is flowing through the kidney. And then finally, the last one that we're going to mention is going to be these that are located here. They're very small, but there's one here and here. There's also one here. The nuclei are slightly resembled here, so I'll erase that so you can see. The extraglomerular mesangial cells. These are important because they have the ability to phagocytotize any filtered particulates from the blood. So it's a form of an immune system defense of any filtered particles that may have entered into the system. So the next part that we'll look at is this glomerulus capillary system. And when we're looking at the glomerulus capillary system, this is the beginning of filtration. So the first thing that we should look at is going to be the endothelium of the glomerulus here. So the endothelium it has a fenestrated, as you can see, the different holes. This fenestrated allows for plasma and dissolved substances to be pulled out into the renal corpuscle. So anything that's pulled out eventually is going to be then turned into filtrate and then go through the proximal convoluted tubule. Since the fenestrations are not that big, we do not see things such as erythrocytes, leukocytes, or platelets being pulled out. The next part of the filtration membrane is going to be the basement membrane. And with the basement membrane, this basement membrane is going to restrict, so this outer covering, this basement membrane is going to restrict the passage of any large plasma proteins to escape also. So that maintains the blood being able to go through the capillary system and then being pulled out through the efferent system without any plasma proteins being pulled out. And finally, the last part is going to be the visceral layer. And the visceral layer of the capsule has these structures called a podocyte. And the podocyte are going to have extensions. So this would be the central nuclei of the podocyte. And the extensions are called pedicels. And the pedicels, their main job is to support the capillary walls. They also interlock with other podocytes. And we can also see the filtration slits inside of the interlocking pedicels. And these filtration slits allow for anything leaving through the fenestrated capillary walls to be pulled out and pulled into the filtration process. So this is going to be an overview. This slide is a main overview of leaving from the glomerulus into the transition into urine production. So again, just for review, what we're going to be looking at is blood entering into the glomerulus capillary system, filtrate being pulled out, and first entering into the proximal convoluted tubule. So there are three different types of filtration, reabsorption, and secretion that's going to occur throughout this system. We just talked about glomerular filtration. It's basically the movement of substances from the blood into the capillary space. And once we get into this capsular space, this is what we're going to call filtrate. We also have a term called reabsorption, and reabsorption is really tricky because you think it's pulling things out of the blood, but it's actually pulling things from the renal tubules into the blood. So reabsorption means we're going to be pulling out any ions, waters, or solute concentrations that were pulled out. If the blood flow, if it needs to be pulled back into the blood, then what we will see is tubular reabsorption. And the last one is going to be tubular secretion. So this means that if an ion or a solute or something was missed, we still have the ability for simple diffusion or through osmosis to allow these substances to move from the blood into the tubular fluid. So again, just to stress, once we're in this tubular formation, what we're going to be doing is either pulling ions and solutes back into the blood or pulling more from the blood. So this whole process, as the filtrate goes through, this is going to be filtrate, and it doesn't really become urine until it leaves the collecting duct. And once it goes into the renal pelvis of the kidney, then we don't have any tubular secretion or reabsorption to occur. So the collecting duct is the last place that we can see these two processes. So this whole system is considered to be a filtrate. It's urine. It's not urine until it is starting to collect into the renal pelvis. So once we get into the proximal convoluted tubule, what we see most often is going to be tubular reabsorption. So the majority of what we see in the proximal convoluted tubule is going to be tubular reabsorption. So we're going to see the blood reabsorbing nutrients that were able to pass through the glomerular filtration, and ions such as glucose, amino acids, or things like sodium can then be pulled back into the blood. So 60 to 65% of the water that enters into the filtrate or into the tubular fluid can be pulled out or reabsorbed through osmosis. It's then going to enter into the nephron loop system. And as we're going into the nephron loop system, we're going to first have the descending limb. And so with the nephron loop system, what we see at first as it exits the proximal convoluted tubule, you'll notice that the descending limb is going to be much thicker. This area is going to be lined with simple cuboidal epithelial tissue. And then the end of the descending limb is going to be a thin, which is going to be simple squamous epithelial tissue. And then as it reverses into the ascending, it's going to be thin and then eventually thick. So this part here, the thin, would be simple squamous, and then the last part of the ascending limb of the nephron loop will be simple cuboidal. So we've gone through tubular reabsorption. We're entering into the descending limb of the nephron loop system. And at this particular point, again, we're going to have tubular reabsorption. And tubular reabsorption is going to occur at the vasa recta capillary system. And the vasa recta capillary system, we're reabsorbing potentially sodium and chloride back into the vasa recta capillary system. Once we go through the descending limb of the nephron loop system, we're now going to be coming up through the ascending limb. And once we get into the ascending limb, we're going to come into the distal convoluted tubule. Again, this originates most often in the renal cortex. It's lined with simple cuboidal epithelial tissue, and it starts in the ascending limb. So this whole region is a little bit thicker, and it's going to be lined with simple cuboidal epithelial tissue. Here we actually have the ability for tubular secretion. So in tubular secretion, we could see potassium ions being pulled out of the peritubular capillaries. We can see hydrogen ions from the blood into the tubular fluid. So this gives the blood the last opportunity to release any potassium or hydrogen that needs to be pulled out. Once we get into this area, the distal convoluted tubule, this is where we have the most effect on antidiuretic hormones and aldosterone. In this particular area, we see antidiuretic and aldosterone increase water and sodium reabsorption from tubular fluid. So that means when we get to this section of the distal convoluted tubule, this is where we see antidiuretic hormones pulling water and sodium back into the blood flow. And of course, any antidiuretic hormone or aldosterone, if you're pulling water and sodium back into the peritubular capillaries, then we're going to see an increase in blood volume and blood pressure. And then finally, we're going to go into the collecting tubule and then eventually into the collecting duct through the renal medulla. So in this picture, we'll just go over this one more time. We see the blood entering into the glomerular capillary filtration system. We see the filtrate leaving into the proximal convoluted tubule, eventually going into the nephron loop, so we can have tubular secretion or absorption, eventually into the distal convoluted tubule. And what this is trying to show you here is just a cross-sectional area of the different cells. But we're in the distal convoluted tubule, and then we enter into the collecting duct. The collecting duct can be considered your last chance. So again, in the collecting duct, we can see antidiuretic hormones and aldosterone increasing water and sodium reabsorption. And what that allows for is if we're pulling water and sodium back into general circulation, then once we get into the collecting duct, we see a more concentrated urine. Once we get into this, the collecting duct is going to travel down, and then again the pathway is going to go through the renal papilla of the medulla into the minor calyx, into the major calyx, the renal pelvis, and eventually out through the urinary bladder and excreted through the urethra.